Introducing the MGF-0416 Flex Total Nucleic Acid Purification Kit with the MGX16 Automated Nucleic Acid Extraction System. This system is designed for the rapid isolation of nucleic acids from various sample types. The kit includes a lysis buffer, wash buffers, an elution buffer, 96 well plates, a magnetic tip comb, proteinase K, and magnetic beads. Ensure all components are at room temperature before use. Before starting, wear gloves and disinfect the working area. Prepare all kit components and ensure they are at room temperature. Addition of binding buffer. First, add 500 L of binding buffer to well 1. Addition of distilled water. In well 2, add 480 L of distilled water. Addition of wash 1 buffer. In well 3, add 1000 L of wash 1 buffer. Addition of wash 2 buffer. In well 4, add 1000 L of wash 2 buffer. Addition of wash 3 buffer. In well 5, add 500 L of wash 3 buffer. Addition of elution buffer. In well 6, add 100 L of elution buffer. Addition of proteinase K. In well 1, add 10 L of proteinase K. Addition of magnetic beads. In well 2, add 20 L of magnetic beads. Addition of sample. In well 1, add 200 L of sample. Load the plates and the magnetic tip combs into the MGX16 device. Power on the MGX16, press the Quick Mode button, and start the run. The MGX16 will proceed through the extraction steps, indicated by the green lights on the device. Once the run is complete, collect the purified nucleic acid from the elution wells. Store the eluates at -20°C for future use. Activate the UV disinfection mode for 1 hour to clean the device. Thank you for watching our tutorial on using the MGF-0416 Flex Total Nucleic Acid Purification Kit. For more information, visit our website or contact us at sales@vitrosens.com.